Hello, folks. This is the live broadcast of the Apologetics Academy, a weekly webinar we run discussing issues that matter, especially relevant to the truth of the existence of God and Christianity in particular. And this evening we're going to be discussing the issue of common descent and evolution. The title is The Common Descent, the Good, the Bad, and the Ugly. And we're joined by our special guest, Dr. Paul Nelson, from Biola University, and who's also a fellow of the senior fellow of the Discovery Institute. There's a number of ways you can participate. You can participate in the chat window. You can also click the raise hand button when we get to the Q&A period after the presentation. You can come up to panelists and ask your question that way. You can also submit questions in textual form with the Q&A button at the top of your screen, anonymously if you wish. And or you can just sit back and listen and enjoy the show anonymously. Also, Dr. Nelson is going to have to leave us, I think, around 20 past. So we've got about an hour and 20 minutes, I think. So we're going to go through this presentation. There'll be time for some Q&A after the presentation. So with that, I'll just turn over to Dr. Nelson. Take it away. Go ahead.
Thank you very much, Jonathan. Thank you again for sponsoring this online academy. This is my second presentation here. It is not part 2 of my ontogenetic depth talk, which I've promised. I will still deliver that, but I thought that it was more timely at this moment to present this talk on common descent in light of some developments in science over the past couple of years. So why use this subtitle, The Good, the Bad, and the Ugly? Well, let me give you an alternative, if that's obscure. After all, not everybody knows 1960s spaghetti westerns. You could call this talk some advice about how to live in 2 different conceptual worlds, that is, within biology, without going crazy. So I'm a fan of Sergio Leone and his so-called spaghetti westerns. This one in particular was the third in the series with the title The Good, the Bad, and the Ugly, starring Clint Eastwood, Eli Wallach, and Lee Van Cleef. Now I'm going to borrow that title for 3 aspects of the theory of universal common descent, which is the canonical geometry or topology for the history of life on planet Earth, where we have the 3 major domains: bacteria, archaea, being unicellular organisms lacking nuclei, the eukarya, in which we nest. We're on this branch here, the animals. All the warm, fuzzy things you see and the big, cold, scary things like sharks on Animal Planet are on this branch. So you can see we're only a small fraction of the total diversity of life on this planet. This theory is universal in the sense that it applies to all organisms, whether extant today or extinct on Earth, rooted in the last universal common ancestor here at the origin. So that's the main character in our drama, but we're going to look at that character in 3 aspects. The first I'll call the good, that's Clint. The second is the bad, and then the last is the ugly. Now, actually, in the movie, this guy's the villain. The ugly character is not really the villain in the story. But I'm going to modify that slightly, where we have increasing degrees of problematicity, if I can use that somewhat awkward word, where the ugly is the worst. Okay, the bad is bad, but the ugly is even more bad in a certain sense, as I'll explain. So how should we understand these different dimensions of universal common descent? Well, let's back up a little bit and note that in July 2018, we stand at a remarkable period in the history of biology. And its features were diagnosed by Thomas Kuhn in his classic, The Structure of Scientific Revolutions. He points out that when dominant theories run into problems, there's a stage at which you have, so to speak, a flourishing of alternative considerations, alternative models. This is how he puts it. In this critical phase, the proliferation of competing articulations, the willingness to try anything, the expression of explicit discontent, the recourse to philosophy and to debate over fundamentals, these are symptomatic of this transition from normal to extraordinary research. And I think that all of these symptoms are present currently in evolutionary theory. I just want to give you very briefly an example of that from Gerd Muller's opening talk to the Royal Society in London at the meeting held there in November 2016, which I attended. Muller is a professor of evolutionary biology at the University of Vienna. He's actually the head of the Department of Theoretical Biology there. And he gave the opening talk on Monday morning at the meeting that the Royal Society convened on the extended evolutionary synthesis. In that talk, he first summarized the main claims or the main propositions of neo-Darwinian theory. He then offered what we could call an indictment of that theory. In particular, he argues that it fails to account for the main phenomena in its domain. Now, he will be addressing, in remarks I'm about to play for you, mainly the evolution of animals, but the critique he presents actually is quite general. It applies right across all three domains of life. And you'll see that what he says fits very well with Kuhn's symptomatology of theory crisis. Now, I'm hoping this audio file will play and that you can hear it. I'll turn up the volume a little bit. What
Genetic inheritance alone accounts for the transmission of selectable traits. And natural selection actually is the only directional factor that is acting on these incremental and slight differences. Note that the majority of these explanations rests on a genetic argument. Really, the synthesis theory is a theory that is focused on variation in populations and on its genetic underpinnings. That is really very important. That's the core of the theory. And what does it explain? Well, it explains very well what it is designed for to explain, namely variation, genetic variation in evolving populations. Makes very good predictions on that. There is, it explains to some extent the gradual variation of in phenotypic characters, explains adaptation of characters, and explains some of the features, the genetic features of speciation. However, what it does not explain are all these complex levels of evolution that I had mentioned at the beginning, such as the origin of these body plans, but also complex behaviors, complex physiology, development, and the fact that not all of the variation that's being generated is actually equally distributed. There are biases in variation. There is novel characters. The standard theory is focused on characters that exist already and their variation and maintenance across populations, but not on how they originate, non-gradual forms of transitions, and all the non-genetic factors of evolution that are involved are not addressed, and actually the theory is not designed for addressing them.
Now, this is an expression of explicit discontent, and it's representative of many such expressions over the past decade within evolutionary biology. So this is the actual slide that, the text at any rate, that, that Muller showed in his opening presentation at that meeting. And arguably, these are the central things that we want a theory of evolution to explain for us. Now, what's interesting is the empirical signal that we could take away from these deficits of the standard theory is nonetheless strictly limited. The question is why? So here we need, again, to step back, take one more step back, and look at a critical logical point that is often deep in the background, if, in fact, one is conscious of it at all. So on the left here, you should see a schematic representation of the two core claims of textbook theory, namely that common ancestry is a fact, that all life shares lineages going back to the last universal common ancestor, and that happened via no design. This is what Darwin is said to have discovered or made quite general in 1859 in The Origin. But of course, the only way this geometry on the left can be an empirical finding, that is something that one discovered rather than an axiom that one has assumed, the only way that this geometry can be an empirical finding is if this geometry on the right is also possible. And in fact, when Darwin set sail on the Beagle in 1831, he thought the geometry on the right, where you have separate descent via design, he thought that was the case, evidentially. Now, of course, he ends up rejecting that geometry. He argues in The Origin that geometry is false. But in order for the geometry on the left to be a discovery, that is an empirical finding, the geometry on the right must be possible. So in The Origin, at page 6, he says, look, the view that most naturalists entertain, which I formerly entertained, is erroneous. That is, separate ancestry via design. He found, Darwin in 1859, found that geometry empirically wanting. But it was possible. It might have been true in light of the evidence. In fact, Darwin says, no, I think it's not the case, and I'm going to argue for the geometry on the left. Now, given the logical possibility that the evidence could have spoken for separate ancestry, it's very important for us to be able to say if the evidence points in that direction. In other words, we should not adopt a geometry of continuous lineages from a common ancestor via no design. We should not adopt that as an axiom, because whatever can be false might also have been true. We need to let the evidence tell us if the geometry that we favor, in this case common descent via no design, if that geometry is false, we need to be able to discover that. Here's what happens in the middle of the 19th century, and we still live in this scientific universe. The geometry on the right, which Darwin rejected on the basis of evidence, is then moved wholly beyond science. We can label it this way: it becomes magic and intrinsically unscientific. Now, this is a very unhealthy situation for evolutionary theory to be in, because it removes from science any theoretical competitors for the geometry on the left. And under those circumstances, all kinds of mischief can occur, because evidence that may indicate discontinuity via design isn't allowed really to speak. And you end up with a very unhappy situation in terms of evaluating evidence, where the theory on the left, the geometry on the left, becomes axiomatic. And that is the ugly dimension of universal common descent. We could summarize it this way: because universal common descent has no competitors within natural science, it lives in constant danger of becoming an axiom. That is, it is presupposed by biological inquiry, but never itself is at risk of evidential challenge. And as I'm going to argue in this presentation, when we come to the question of evo-devo, but of course the problem is quite general. I can give you an example from molecular systematics, molecular phylogenetics, and the Q&A if anyone's interested. That's quite striking. This danger of becoming an axiom is quite real. And in fact, it was present in the immediate post-Darwinian period. William Bateson was born 2 years after the publication of The Origin, so he came of age as a scientist in the immediate post-Darwinian period. And in his great work, Materials for the Study of Variation, published in 1894, he says, I'm going to assume that descent, meaning common descent, is true. Now he says, this has never been proved. Has evidence in its favor, but really, he says, it finds its support not so much in direct observation as in the difficulty of forming any alternative hypothesis. So you can see, even at this early stage, in the decades right after The Origin, theoretical biologists are beginning to place alternative geometries actually beyond the reach of natural science. And I think, as I'm going to argue, that creates a very unhealthy situation for evolutionary theory, because it lives then in constant danger of being held axiomatically. So here is what I would describe as a healthy situation for the theory of common descent, where the double-headed arrows indicate that traffic, so to speak, is possible, moving in all directions. You could have new evidence arise that may generate a research problem, or it may directly challenge common descent. And movement among the 3 points of this triangle is possible in every direction, because common descent has not been privileged over the evidence, and research problems that fail, a research program that fails, may itself challenge common descent. In other words, there's traffic moving in all directions. In fact, this doesn't accurately describe how most biologists actually evaluate evidence. This is a much more accurate picture, where common descent is privileged with respect to both what the evidence might tell you and what research problems you have, and it tends to be insulated against empirical challenge. And I'll illustrate that again in a few moments using examples from evo-devo. Now, in this situation, observational anomalies generate research problems. They don't threaten the truth of undirected common descent. Again, the danger is that common descent may be rendered axiomatic if you hold it this way. So Whewell, a philosopher and historian of science, a polymath actually at Cambridge, who influenced Darwin in Darwin's thinking, in his great work, Philosophy of the Inductive Sciences, he has this beautiful aphorism that I think should be on a poster on the wall of every scientist's lab. Take with him or her out into the field, and that is the following: there is a mask of theory over the whole face of nature. And that's unavoidable in a certain way, because we have these instruments, theories, that we take out into nature, but they can end up covering the face of nature in a way that's really problematic. Now, typically in any talk I give, I tell the audience there's only one slide that you have to remember, all right? And this is the one. If you don't remember anything else from this talk, remember this slide. Universal common descent acts as a perceptual filter on the evidence to a degree that few biologists fully understand, including, surprisingly, intelligent design theorists. So we begin with the data, with the observations, and they are filtered through the assumption of universal common descent. That then yields what is often described in the molecular phylogenetic literature, for instance, as the phylogenetically informative data. Now, that will feed back on itself. And many biologists are not actually conscious that they are doing this, that when they look at observations, when they begin with raw data, they filter it through the assumption of universal common descent, yielding what they regard as phylogenetically informative, which then feeds back on itself. And if you don't have a way of breaking that loop, namely of knowing the circumstances under which universal common descent could be challenged by data, you have effectively made that theory a filter or an axiom. And that I think is problematic, because we don't want to hold the theory axiomatically. Although, as you'll see at the end of the talk, some biologists, in fact, do consciously hold it as an axiom. So let's take a problem like this, the origin of the animal phyla. All right, fascinating puzzle. We can pose the question, do the animals share, do the bilateria, and there were actually some other non-bilaterian phyla exhibit on those two slides, but let's say, do the bilaterian phyla share a universal common ancestor? Now, there are two possible, not a universal common ancestor, but a metazoan common ancestor. There are two possible answers to that question, yes and no. No has become labeled as creationism, right? It's off the table, it's out of consideration. Magic, it's gone. So we have yes. Now, yes can lead us to standard evolutionary theory. And again, the question would be, well, yes, that's adequate. Standard evolutionary theory, textbook theory, is adequate to explain both necessary and sufficient to explain the origin of the animal phyla from a common metazoan ancestor. So it's just a question of applying the theory we have to the problem and sorting out the details. So Douglas Fatuma, in a paper published just before the Royal Society meeting, said, look, evolutionary theory gives a plausible account of all the phenomena in its purview. He says, I don't know of any macroevolutionary phenomena that are inconsistent with existing theory that would require us to reject one of its principles as simply false. And actually, Douglas Fatuma was actually at the Royal Society meeting, sitting in the front row for, I think, for the whole thing. Now, you can answer that question downstream of the yes, the first yes. You can answer it no, which was, in fact, the motivating answer, I think, for many of the speakers at the Royal Society meeting. They said, no, textbook theory is not adequate. But notice that both the yes and the no here are within the domain, theoretical domain, still of common descent. And common descent, therefore, sets the disciplinary boundaries for the questions worth asking and their acceptable answers. So what I'm going to do is challenge the big box, the big red box. I think that we need to be able to overcome our fear of creationism, if you want to call it that, and say, yes, we can say no here. And in saying no, or in answering no, we actually end up with all kinds of interesting implications, such as, why do mutagenesis experiments in animals show at best the losses of structures? Why are discontinuities in the history of life and in the distribution of extant species so pervasive that Eugene Koonin, calls them a Big Bang pattern? Why has it been so difficult for standard theory to solve the origin of body plans and so on? This is a largely unexplored territory, although intelligent design theorists are moving into it with dispatch right now. But there's more. There are data that we should see if, yes, we're true, that we actually don't observe. In fact, we never observe them, as I'll explain in a minute with respect to evo-devo. Now, I would argue this is not simply a question of incomplete sampling, that in fact what we have is something closer to a genuine anomaly in Kuhn's sense, okay? So one of the challenges the intelligent design research community faces is to peel back this obscuring mask of this be magic, all right? Get rid of that so that we can begin to answer the question, look, if the geometry on the right were actually the case, what should we expect to see? And I think in the past a few days, Winston Ewart's proposal of dependency graphs provides possibly an avenue for answering that question. But I think for much of the biological community right now, that is still in place. And any alternative geometry to the one on the left, the continuous geometry of common descent via no design, is still considered to be magic. All right, that brings us now to really, I think, an important consideration, and that is this. What are the other good aspects of universal common descent? And it does have many, right? Well, the first is, at a deep level, an erroneous theory is still better than no theory at all. Francis Bacon, during the early scientific revolution, put it this way: the truth emerges more readily from error than from confusion. Error has structure, confusion does not. Even a false theory will teach you things about the world because it will point you in certain directions where you can go out and look, and then nature talks back and you end up learning things. So when universal common descent is coupled with the rest of our biological knowledge, it does give a testable framework for doing historical and comparative biology. In that respect, it rises to what I would call the dignity of error. That because it does tell you things about the world that might turn out not to be the case, you are going to learn things. But it gives you more than that. Excuse me, I'm going to skip that slide. Actually, let me go back here. If this geometry is true, in particular, if Luca existed at the root of the three domains, all by itself, that will give you predictive power. All right, because evolution is not an unconstrained process. So somehow we get Luca up and running, the first cell, And it's going to be a pretty complicated creature, probably with about 300 proteins, dozens of large RNAs, very large structural RNAs, for instance, in the ribosome. The central features of the biology of that cell, because of their functional necessity, will be inherited with very high probability by all of its offspring. Now, that ends up giving you all kinds of predictions that you can make about what you should expect to see as you climb up into the tree of life. There are viability constraints on evolutionary pathways that allow you to predict that functional elements ought to be retained as you climb up through the branches. This has enormous predictive power. And the fact that the predictions in many cases have failed, I don't have time today to go into the details, the fact that many of those predictions have failed is teaching us something about the history of life. So you get a time scale with universal common descent. You get an historical narrative of this monophyletic branching pattern and so forth, and you get process expectations. All of this yields predictions. And in that respect, universal common descent is a powerful tool for learning. But let's go back to this problem. Because it doesn't have competitors, it ends up running the risk of becoming a filter on data and eventually being held axiomatically. So now I want to look at the bad aspects. And this will be the longest part of my talk. I want to move as fast as I can so that we'll have plenty of time for Q&A before I have to leave. So let's look at evo-devo. Although the mode of reasoning that I'm going to examine is quite general. You can find it in abundance, for instance, in the field of molecular phylogeny. As I said, I have an example that we could discuss in the Q&A if there's time, illustrating that. So let's look at the problem of the origin of animals. Now, even at its zenith in the mid-1960s, when the modern synthesis really had no competitors, even within evolutionary theory, punctuated equilibria was yet to be born, structuralism was yet to be born. The modern synthesis was held by, I think you could say, within the Anglo-American sphere at any rate, all the leading investigators. Even at its zenith, people like Conrad Waddington at the Wistar Symposium were expressing discontent, unhappiness with the theory. So at the Wistar Symposium, Waddington said, look, the whole real guts of evolution was how do you come to have horses and tigers and things lies outside the mathematical theory, meaning population genetics, which is at the core of neo-Darwinian theory. In particular, the problem of the origin of the major discontinuities among the animals, which we'll be focusing on, was unsolved. And it was unsolved because development, the processes of development by which these body plans are constructed, was never properly integrated into neo-Darwinian theory. And there's a reason for that we'll come to in a moment. So in 1987, I was a graduate student at Chicago, and Wallace Arthur came to Chicago from the UK for a sabbatical stay. And at the time, he's an evolutionary biologist trained in both population genetics and developmental genetics. At this point in his career, he was becoming increasingly unhappy with the theory that he was taught and that he was teaching and trying to apply. And in this publication, he said, look, we don't really know, or we don't really have direct Darwinian evidence, direct evidence for a Darwinian origin of body plans. Therefore, we really have to admit we don't know how body plans originate. And from that point on, late 80s, as you track his publications into the 90s and further, he is increasingly outspoken about the need for an alternative account of the origin of large-scale form. Eric Davidson, who died in 2015, over the last 2 decades of his career was very outspoken about this, that the kinds of variations that you could observe within natural and experimental populations, for instance, in Drosophila or his own model group, the purple sea urchin, were not relevant to the problem of the origin of large-scale form. He compares them to apples and oranges, right? He says we're going to need a new theory for the mechanisms of body plan evolution. And again, these scientists are representative of a much larger body of opinion about this problem. And it tends, the problem tends to center on what kind of variation we need to modify, A, to construct, but B, to modify body plans. You could call this the mystery of the missing deep variation. And you could summarize it this way. This could be put, it's a little long for a bumper sticker, but you could summarize it this way. Mutations expressed early in development affecting body plan formation are those least likely to be tolerated by embryos. This is the clear signal that comes to us from Drosophila, from C. elegans, from mice, from zebrafish, from all the major model systems of evo-devo. Embryos do not like their early stages tinkered with, for very good reasons, as we'll see. So I remember when this paper was published in Nature. I was a freshman studying evolutionary biology at Pitt, and there was great excitement around this paper and other papers from Nusslein, Volhard, and Wieschaus, because with their Germanic thoroughness, using saturation mutagenesis in Drosophila, they really hit just about every relevant locus in that genome bearing on the development of the flies. And they were able to reverse engineer how Drosophila normally developed. In so doing, locating the genes and their protein products, DNA regulatory elements almost entirely, that control the process, okay? So as I said, it's a type of reverse engineering. What they did is, if you think of development in Drosophila as an expanding cone of decisions, starting from the fertilized egg, you have this expanding code of cell number and cell differentiation within which all kinds of decisions are occurring. Committing cells to their terminal tissues, to their terminal functional roles. You're going to be a neuron, you're going to be part of the gut, you're going to be a wing, you're going to be a leg, and so forth. And by disrupting genes at different intervals temporally within that cone, Nusslein, Volhard, and Wieschaus said, well, if we break this enough, we can figure out how it works. Okay, so here's a little cartoon showing the cascade, the developmental cascade of decisions in the early stages leading to the larval form in Drosophila. Again, I don't have time to go into all the details, but they discovered this. They, of course, and other people discovered this by painstakingly breaking the process over and over and looking at which decisions depended on which, working their way right back to fertilization, of course, in later work, ultimately to how the oocyte was itself set up in the mother's egg chamber. Now here's a figure from their 1980 paper where you can see that you have on 4 columns. On the left-hand side of each column is the wild-type form of the larva, and the regions colored in pink are going to be those disrupted by the mutations they induced. On the right-hand side of each column, you have mutant embryos that they were able to recover. Now, the mutant embryos are all dead. All right, there's dramatic change in their form, but they're not going anywhere. And it's this paradox, the consequence that inducing large-scale change in embryonic form pushes you off of the normal trajectory of Drosophila development into a lethal zone. There's an evolutionary conundrum that follows from that. It's not hard to understand. Given the causal dependencies, that crashing of the embryo is exactly what you should expect, okay? A mutation that's expressed late in development may affect only a few hundred or a few thousand cells, and it may be tolerated by the fly. So you might change bristle number, you might change pattern of venation in the wing. In other words, it would be tolerated by the adult form of the fly, whereas a mutation expressed early in development is overwhelmingly likely to be catastrophic because cell number is very much lower. The consequences of that mutation will tend to ramify throughout the whole decision tree. And in fact, this is what Nusslein, Volhard, and Wieschaus found. All right, and it ends up being a 3-point argument. The first point we know from development, not all animals, but most animals begin their existence as a single cell of fertilized egg. The earliest stages in that process determine what follows. If we're going to change the adult form of the animal, we have, because its adult form is first set in place at the very earliest stages. We're going to have to modify those via mutation if we want to evolve the phenotype of the adult. So you're going to have mutations that are expressed here. And now the paradox, those are the ones least likely to be tolerated by the embryo. And this is the clear signal that comes back to us from all the model systems of evo-devo, not just Drosophila, but really everything else. Now, this problem has been known for many decades, as I'll show you in a few moments. When I was a student, it was already well understood, and it cannot be solved within the neo-Darwinian framework. One of the reasons for the Royal Society meeting was the insoluble character of this puzzle for standard theory. And the consequences, of course, for the origin of the animal phyla are just as severe. If we suppose that there was a common ancestor to the bilaterian phyla, call it Urbilateria, it would have undergone a developmental process, probably a relatively short developmental process. Maybe it had only a few thousand cells. But to change the form of Urbilateria, which is illustrated here, this is just kind of a cartoon balloon. There's very few features here. There's a gut, but very little else has been illustrated. The illustrator here has provided some sense organs and so forth, a cuticle. There is going to be some degree of differentiation even in this very simple animal. To modify its form, you would have to change its development, and the same problem would recur. All right, so there's your bumper sticker. It's a little long for a bumper sticker, but that's the problem. Take that to any evo-devo meeting, and you'll sustain hours of discussion. Now, someone might say, well, look, Paul, you've just got a bad sample, right? Somewhere beyond the boundary of the mutagenesis experiments you've shown us are the lucky mutants that are going to make a go of macroevolution. And remember, natural selection is a probability amplifier. It thrives on the occasional rare but lucky event. And I've actually had this response from scientists at evo-devo meetings. The problem is there's no reason to believe it's true, okay? And the reason we shouldn't believe it's true is the same reason, in a sense, that you should not put money into these machines. If she starts with $1,000 and keeps feeding all of her winnings into this machine, it's mathematically certain that at some point in the future, the casino will have all her money and she will have none, all right? And there's no doubt about that. It's mathematically certain. So in a sense, the argument that I've been given is the gambler's fallacy applied to evolutionary biology, right? The data we already have in Drosophila, in the case of Drosophila, over 100 years of mutagenesis tells us if you want to fly at all, you have got to stay somewhere in the neighborhood of the wild type. And there's no reason to think things will be different. So let's look at mice. Now here's a table from an experiment using a powerful mutagen in mice. And if you go through the table, and this will be the case really for any model system you look at, zebrafish, it doesn't matter. The mutants that are there, either they've got some defect that they're able to tolerate, but they are less fit than the wild type, or they're hopelessly damaged, okay? And these mice aren't going anywhere, right? The mutations that were induced are so severe that you can recover the embryos, but there are no stable populations with these morphologies that you could have breeding populations of mice. There's no reason to think your luck is going to change, all right? It's the gambler's fallacy. The champagne will never be delivered. So, skip that. This problem has been known for a long time. So when I was a student, first Reagan administration, this is a long time ago, okay? John McDonald, who was looking at this puzzle, summarizing 20 years of research. So he's looking at findings from the 60s and 70s, but really going back through the whole history of the modern synthesis. He says, we've been confronted now with a great Darwinian paradox. And I'll summarize what he's saying here. Those loci, the genes in populations where we observe variation in both natural and experimental populations appear not to lie at the basis of major morphological change. We don't see those variations. The reason we don't see them is the embryos won't tolerate them. Major adaptive changes of the sort needed for macroevolution apparently are not variable within natural populations. Now, Davidson, in a paper in 2011, put it very bluntly. He said, textbook theory rests on a set of assumptions that what he calls basically counterfactual. That micro and macro evolution are qualitatively different, and the evolution of body plans, right, cannot be derived, or our understanding of the evolution of body plans cannot be derived from what we see in the experimental populations that we are looking at today. Something very different is needed. And he ended his career looking for that alternative theory. So to stay on schedule, I'm going to try to go through these next examples very quickly. We can look at this with respect not just to evo-devo, but to what we would predict about the history of the animals using Drosophila as our model system to do so. So when I came to Chicago, this cascade was being worked out in great detail. And you can generalize from this and other model systems and develop a theory, which my advisor at Chicago, Bill Wimsatt, did, that he called generative entrenchment. Now, here is a visual metaphor for animal development that Wimsatt developed. It's really quite elegant. Think about this like a bicycle lock, okay? It wouldn't be a practical bicycle lock because it has too many wheels, but here is a bicycle lock with 10 wheels, each of which has 10 settings. Now, to solve this lock by random perturbation of the wheels would take you a long time indeed, 10 to the 10 combinations. The only way the lock will open is if all the wheels are put in the right position, and it's going to take you a very long time indeed to solve it. So Wimsatt calls this a complex lock. Actually, he borrows this simple and complex terminology from the Nobel laureate Herbert Simon. By contrast, here is a simple lock, what Wimsatt calls a simple lock. Now, this lock has 10 wheels, but each individual wheel has a reporter function. In other words, when you get the wheel in the right position, you'll hear a little click. Now, a child could solve this lock in a matter of moments because you turn the first wheel, you hear a click, turn the second wheel, keep turning it, you hear a click. Make your way through the 10 wheels, you'll be done in a matter of moments. This would not be a practical bicycle lock because any thief could open it with very little trouble at all. Now, Wimsatt, in a sense, forms a hybrid of these two locks in a very interesting way. He says, look, we can think about development in animals going from left to right as a simple lock. In other words, if your early cleavage stages work out okay, you can move to the next stage and so forth, and you'll solve your trajectory here of development easily if each stage ends up being functional in the right position. He said, but going the other direction, it's a complex lock because wheel 10 can only be solved if wheels 1 through 9 are already in their correct positions. Okay, so there are relative degrees of functional dependency here such that these early positions are what Wimsatt calls generatively entrenched with respect to the later ones. And actually, this turns out to be a very nice model for animal development. And Wimsatt actually then develops an entire theory that he calls generative entrenchment for these causal asymmetries or functional dependencies in complex systems. All right, you can apply that. He first derives it from development, then you can apply it back to development and ask, what should we expect to see as we look at the development of these different animal groups? All right, they've got ontogenies that they're going to undergo. What should we expect to see as we look at them? Well, what you would expect is something like this if common descent is true. So here's a little cartoon showing an evolutionary lineage starting here with population A, and we get time going from past to present, and disparity is increasing. And if A develops by a relatively simple ontogeny, A to B to C, where A is more deeply entrenched than B and C, we will tend to add stages to the end of that developmental pathway, such that when we get to C, and we've elaborated this developmental pathway, A now, excuse me, is going to be much more deeply entrenched here than it was here. Even though it is entrenched here, it will be even more so here because you've added stages that are dependent on A going right. Now, this prediction, actually, you can find in the literature well before actually Wimsatt offered it. It wasn't called generative entrenchment, but the same idea, for instance, can be found in a paper in 1971 by Britton and Davidson, where Davidson says, we would expect that regulatory programs active earlier in development of any animal would have been elaborated at earlier stages of evolution. Why is that? Well, because of this pattern. If we compare A to C, they are going to tend to, if they are related by common descent, they are going to tend to share these early stages. Davidson says therefore, the later stages would be, excuse me, as the later stages evolve, the earlier stages will be more or less fixed, and it's unlikely they could be supplanted. They would have been elaborated earlier in evolution and should be more widespread phylogenetically. Okay, so given David's argument, if we do a little cladogram here of these 6 species, this is the distribution we should expect to see, where species F is the most derived, the last to evolve. Species A is the least derived or most primitive, the first to evolve along this cladogram. We would expect, if these 6 animal species share common ancestry, that their development should be conserved at its earliest stages. It's a clear prediction that flows from common descent, plus what we know about animal development. So we can put in 6 of the major model systems of developmental biology with this branching pattern. Mammals being the most derived, these early groups less so, and we should expect conservation of the early stages of development in these groups. All right, now, Drosophila and C. elegans cluster together in the group the Ecdysozoa, the molting animals. One would expect, on the basis of the prediction we just derived, that their early development should be conserved. In fact, it's not. Dramatically not so. So here's the earliest stages of C. elegans development, where you have the fertilized egg here branching to 2 daughter cells, which then set up additional branching lineages. Notice the architecture here. The very first event is cleavage, cell division, setting up asymmetric lineages. When we look at Drosophila, there aren't any cells until fairly late, right? So we have a single large cell here, the syncytium. The nuclei begin to divide here in the center of that cell. They migrate to the periphery of the syncytium. Cell membranes then arise among those or between those nuclei. There's really no cell division to speak of until very late in the process. These architectures could not be more different, even though Drosophila and C. elegans cluster together in the Ecdysozoa as putative kin. If you look at Davidson's publications, between the 71 paper and what he begins to publish in the late 80s, early 90s, he totally shifts his perspective. And he says, there's no reason to think that animal development should be conserved in its earliest stages. Now, what happens in his career between 71 and these papers is he writes a textbook on comparative animal development, moves out away from the purple sea urchin to look at other groups, and he says, look, they are about as different as can be. Early development in the animals, excuse me, is not conserved. Now, at Chicago, at the same time that I was there, Nick Rasmussen was another graduate student working with Bill Wimsatt. And Bill said to Nick, let's take my developmental lock generative entrenchment model and apply it to Drosophila and see if we can generate predictions about the distribution of genes and their protein products as we move out from Drosophila into the Diptera, into the Insecta, into the Arthropoda. What should we expect to see? So Nick does this, he applies the model, and he says, look, younger genes will be less entrenched than older genes. And the genes and proteins at the deepest levels of entrenchment within the Arthropoda should be the most widely shared. A strong prediction, right? Because the downstream consequences for introducing a novel regulatory element into one of these decision trees, either it will be integrated into the existing tree or it will be catastrophically deleterious. Those are about the only 2 options you have. So Nick says, look, the elements that are most deeply entrenched in the Drosophila cascade should be those most conserved among related species. So here's the prediction. There's Drosophila development there, and it nests within the diptera. The diptera nests within the insecta and the arthropoda. And the prediction is, as you move out into the arthropods, the regulatory elements most deeply entrenched in Drosophila development should be conserved to larger clades. And in fact, that was exactly not what was found. Okay, so here are the Drosophilids, all right? And you'll notice they have bicoid. Bicoid is a key regulatory element setting up the banding pattern and the anterior, posterior, during the period when the syncytium is where the action is, so to speak. Bicoid, translated from a messenger RNA put in place by the mother, is actively transported down from the anterior to posterior poles of the syncytium, and it sets up the banding pattern that will enable the later stages of the cascade. It's present here in Drosophila. It's present here. It's present here. But notice that there is no bicoid, right? A next door neighbor relative to Drosophila doesn't have it. It's not present earlier. In fact, these regulatory elements are distributed like Christmas tree ornaments. There's no phylogenetic logic here. In other words, the prediction that Nick made, which was beautiful and clear, turned out to be exactly wrong. Here's the problem. When we couple common descent with everything else we know, we get predictions we can check. In this case, when we couple it to our knowledge of how development works, we get, as you've noticed from just the past little bit, clear prediction of embryonic conservation. What happens when this fails? Something over here has got to pay the price for that failed prediction. And the question is, why is common descent protected when the predictions fail? Okay, I'll give you another pair of examples, and then we should finish a little bit after 3. So here's a parasitic wasp that is a relative within the insecta of Drosophila. Now, our biological knowledge, our background knowledge, would lead us to expect that early changes early in development, would occur rarely because they are lethal, right? Or highly deleterious. So the authors working with this parasitic wasp say, well, look, if ancestry, evolutionary descent is the primary factor driving patterns of early development, we would expect that insects in the Hymenoptera would look much like the honeybee. So the honeybee shares a geometry with Drosophila, where you have a single cell, the oocyte, where the axes, dorsal, ventral, anterior, posterior axes are being set up in the mother's egg chamber. She's pouring information in to set them up, and that what you would need is an open space for the active transport of a morphogen like bicoid from the anterior pole or nanos from the posterior pole to set up the banding pattern that will enable development. In fact, what we see in this wasp is complete cellularization. It's just amazing. So you can see that they've lit up these cells with a fluorescent marker, and these are cells that are impermeable to transcription factors or morphogens like bicoid, because they've injected a dye here into this blastomere, and it's not diffusing. It is stuck in there. And the molecular dimensions of the dye are smaller than those of bicoid, all right? Complete cellularization, as we see in that wasp, would disrupt, here's my little cartoon showing it, would disrupt the motion or the transport of that morphogen. It's a major puzzle how that could have evolved from a common ancestor where a syncytium is the ancestral form, okay? So they just point out here, we injected dyes into the blastomeres, they stayed there, meaning this wasp is developing in a way that Drosophila, the patterning hierarchy could not work because you could not have diffusion or active transport of key regulators like bicoid. If the dye can't get out, bicoid can't get out either. But this pattern is quite general. So here's a midge, Chironomus, that was worked on here at University of Chicago. And in a very interesting paper published a couple of years ago in the lab of Schmidt-Ott, they looked at the regulatory elements that correspond in Chironomus to bicoid in Drosophila. And what they found is that instead of bicoid, which is a transcriptional activator of anterior genes, Panish is a repressor. And its logic is completely different. It's actually a different protein. So let me just put on my cheaters so I can see. That midge is over here. And you can see it doesn't have bicoid present. And this phylogeny is a figure from their paper. And they say, we don't find evidence of Panish, of this key regulatory element in other dipteran genomes, even though it's in some close relatives to the species they looked at. They said, this suggests a recent origin. And their bottom line, our study shows that the mechanisms of anterior-posterior patterning in insects are more labile than previously acknowledged. Well, this adjective really is misleading. I have to be blunt. There's no experimental evidence, none, that you can disrupt these key early acting regulatory elements in insects. What they mean by more labile here is that they had observations showing that at some point in the history of these species, these regulatory elements were fundamentally different. All right, that's a very different claim from saying actual insect development is labile. There's no experimental evidence for that. This is common descent in action. I would say I have great respect for this investigator and for these results, but this shows you the role of common descent in skewing the interpretation of data. Because when this fails, what you could say is we actually know a great deal about this, and it's common descent that's not true. But investigators rarely make that move. And this, again, is the ugly dimension of universal common descent. Now, we've been talking just about the common descent of the animals, but the problem is quite general. If you do this over and over, let me go back here. If when a prediction fails, the cost is not paid by common descent, but you say, well, we have a new research problem. How can key regulatory elements and developmental pathways be swapped in and out without disrupting the development of the species? This ends up becoming an axiom and insulated from observational challenge. It might not be true. This theory might not be true, and we need to be able to answer the question, if a failed prediction arises here, how can we get over here to ask questions about this? Okay, this ends up being the ugly aspect of universal common descent. And this circle, to give it a name, becomes highly problematic because this could be false. It should not act as a perceptual filter. Now, some biologists say, forget it, adopt it as an axiom. Ronald Jenner, in a paper a little over 10 years ago, said, ever since Darwin, we have understood that evolution, we have understood evolution is descent with modification. Consequently, no degree of modification can be used as evidence against common descent. It's genuinely an axiom for him. Darwin made it very clear, Jenner says, that no amount of difference between organisms could impact our decisions about genealogy. All right, you can do that if you want. You can put common descent like a queen on her throne, and she's never challenged by observation. I don't think many biologists do that. If they become conscious of the fact that they are privileging common descent as an axiom, they stop doing it because they want to be able to say this theory could be challenged by observation. Kenneth Weiss at Penn State, in a paper with the title, "We Hold These Truths to Be Self-Evident," he said, look, we insist on a point source for the whole of life, all right? Somehow that first cell got up and running, but we forbid that process to happen at any other time. And he says shared ancestry is an inviolate assumption of evolutionary theory. Again, consciously, explicitly making it an axiom. You can do that. I don't think it's wise because this geometry might not be true. And not only the universal aspect of it, but particular later branching events within it might be false. We need to be able to know how that happens. So I'll skip this for lack of time. I want to end at 3. Common descent should not be privileged over the rest of our biological knowledge. This, right? Everything else we know over here needs to be able to challenge that geometry. All right, so it's 3 o'clock. Here's my little Swiss pocket watch. And we've got 20 minutes for Q&A. Thank you very much for being so attentive.
Awesome, thank you. Do you want to unshare your screen just so we can see your full picture? Just at the top of your screen there is a button. Great, thank you. This is the interactive portion of the program. If you want to raise a question, then click the Q&A button at the top of your screen or click raise hand. I find that really fascinating. I love developmental biology. It just screams design. I find it amazing that people can actually study molecular or developmental biology and retain their naturalism. But on the topic of common descent kind of being insulated from criticism or falsification, I've found quite interesting reading papers on de novo gene birth, where you pull up just a standard review article on de novo gene birth, where you have a gene which just comes into existence without predecessors, and the papers will almost uniformly say somewhere in the paper, it used to be thought that genes from scratch was almost impossible in evolutionary terms, but now we realize actually it's really easy for evolution to produce, because look at all these examples of that. So it's just funny how it's kind of circular reasoning. They won't question the paradigm; they'll use it as just further evidence that confirms the efficacy of Darwinism.
Well, Francois Jacob in 1977 said the antecedent or the prior probability of the de novo origin of a functional protein from random DNA was effectively zero. And as you said, the discovery of orphan genes and related data have stood that on its head. And I'm very interested. I mean, half of me is a biologist, half of me is a philosopher, and the philosophical half of me is very interested in how rapidly opinion can be reversed in the face of novel data when you insulate common descent from challenge.
Mm-hmm. Okay, we have a couple submitted questions. Let me read those. Eric Anderson says, Paul, was the problem of changes early in the developmental process explicitly noted and understood early by some of these scientists who express concern, or did their concerns tend to be based on more general analysis of the limitations of mutations and the mathematical odds?
I think actually there were at least two streams of thinking that led to the problem of early acting body plan affecting mutations. If you look at the major work of Ronald Fisher, one of the founding documents of the neo-Darwinian synthesis, he very clearly points out that macromutations are possible and have been observed, but he adds they are not good candidates for adaptive evolution because they tend to be either lethal or overwhelmingly deleterious. Mm-hmm. And he draws an analogy with a microscope. I actually have a microscope here in my office, but I won't go get it. I can do it with my hands. Imagine the various parts of a microscope, and you're trying to bring your sample into sharp focus. Fisher says it's very unlikely that what he calls a major derangement of any of the parts of the microscope is going to improve the focus of the image. He said, and he actually draws a mathematical analogy. He says that we can associate probabilities with these derangements. It's overwhelmingly likely, he argues, that any major derangement is going to move you away from sharp focus into something that you can't actually see. So he takes that analogy from microscopes and goes back to organisms, and he says they exhibit the same kind of fine-tuning, if you will. He says, and the experimental evidence bears this out, that macromutations, even at the time, this is the late 1920s, it was already well established that major changes tended to be overwhelmingly destructive. So you have these two streams, direct observation and theoretical analysis of what happens to complex systems when they're perturbed, that lead neo-Darwinians to say if evolution occurs, it must occur with small-scale variation over long spans of time. And that generalization isn't really challenged until the early 1970s, when you have a generation of paleontologists like Stephen Gould, Niles Eldredge, Steve Stanley, and so forth, who, looking at the fossil record, say, we see discontinuities in the paleontological signal that look to us to require macromutations. So it's only really not until the mid to late 1970s that theoreticians begin to challenge that neo-Darwinian assumption.
Joe Miller asks, what is the impact of epigenetics on this theory of universal common descent?
Well, I would say at this point, it's still fairly limited because almost without exception, investigators who are looking at epigenetic phenomena will still assume that the species that they're looking at share common ancestry with other animal groups. I can't speak to epigenetics in plants. I don't know anything about it, but I think without exception, every investigator I know who is looking at epigenetic phenomena in animals will say the animals share a common ancestor at some point before the Cambrian that was itself a developing organism. So all the epigenetic phenomena tend to be subsumed within this geometry or topology of common ancestry. It's really not until you're able to say, I'm going to peel back that layer that says that alternative geometries aren't magic, that one can begin to ask the kinds of questions that I would like to ask.
By the way, are you planning to write a review paper on this topic? Because I think it would be really good.
Yes, in fact, Steve Meyer and I have for a couple of years wanted to do a pair of review papers, one of them looking at this puzzle, but another one continuing to develop the ontogenetic depth idea and publish those probably in biocomplexity, although we would love to get them into another journal if possible. Nothing wrong with biocomplexity, but I like to take my case as far as possible to my evolutionary colleagues.
Yeah, absolutely. Zach says, hi, Paul. What is the highest level at which you think common descent is true as a general rule? Example, common descent is only true within the species, genus, family, order, etc.
Very good question. I actually gave a shorter version of this talk to a discussion group earlier this week. The same question came up. The question cannot be answered sitting here in my study, right? Answering that question will require very hard work over a long period of time. Some of it has already begun. But having said that, I would say that I think there is compelling evidence for common ancestry in some groups up to the level of family. And you can see this in hybridization matrices where along the horizontal and vertical axes, you lay out an array of species, and then you score where successful hybrids have occurred among those species. And what you get in the center of the matrix is a very interesting cloud of fertile hybrids, all right, suggesting that the diversification that has occurred into species may be traceable back to an ancestral form that gave rise to these species. And just recently, in fact, just a few months ago, there was a beautiful paper published on the really surprising genetics of a number of cetacea, where it looks like they diversified from a common ancestor. And these are good bona fide species today will not hybridize under normal circumstances. But if you look at their genetics, it looks like they branched from a common ancestor. So I would say, show me the data, show me hybridization data, show me comparative genetic data. But really, Zach, the question can only be answered by very, very hard work on model groups. You really can't do it. You really can't do it sitting in your study. But I'm willing to look at any case. And I would say, where do we see hybridization begin to break down? What kinds of variations are tolerated? So I would start at the level of species and genera and begin to climb up into the taxonomic hierarchy and say, do we find discontinuities that cannot be bridged by known genetic processes, known evolutionary processes? And as you tend to climb up in the Linnaean or in the taxonomic hierarchy, these discontinuities become more and more striking, especially above the level of family.
Right. Similar to the question I asked, but Eric Anderson says, fantastic presentation. Have we prepared a paper on this? What is the next step and how can we help?
As I said, Steve Meyer and I have been planning to do a pair of papers on this, maybe more than two. The challenge, those of you who have watched the presentation, is journal editors and referees get very, very skittish when you raise questions about common ancestry, because that fear of the C word, that fear of creationism comes crashing in on them. And they say, well, look, you're getting out to the boundaries of what is rationally permissible within evolutionary biology. And I don't know if you guys know, but I tried to encourage Winston Ewart to take his dependency graph proposal to the best mainstream journal he could find. And Winston very politely listened. And then he said, no, I want to make my full case for intelligent design with respect to this proposal. So he published it in Biocomplexity, where, yes, he had to go through refereeing, but he wasn't under these constraints that mainstream journals apply. So maybe there's a version of this that could make it into Quarterly Review of Biology or Bioessays or evo-devo, where we soft pedal the intelligent design implications. Unfortunately, to do that, we're going to have to back off challenging the common descent of the animals. But I want to be as intellectually free as possible, so it'll probably end up in Biocomplexity.
Yep. I haven't actually read Winston's paper yet on the dependency graph. I just haven't had time. But I don't know if you could summarize it like really in a few minutes. Probably not. But do you want to just, for those that aren't familiar with the concept, just give a brief explanation?
Winston was originally motivated to look at the question of the apparent nested hierarchy of genes and proteins in the three domains, where, for instance, universally it looks like you have ribosomal proteins, actually ribosomal RNAs that are shared by all life on the planet. And then again, as you climb up into the tree of life, you get increasingly restricted sets of genes and proteins appearing to diagnose particular clades. So as you climb up the branch leading towards mammalia, you will see universal, eukaryotic, metazoan, what's next? Chordate, begin to restrict the tree in smaller and smaller clades, and you'll see clusters of genes present at those branching points. It looks like a nested hierarchy. Winston said, I see the same pattern in software, but I know what's going on with software. You've got modules that everybody uses. They tend to be borrowed from one piece of software to the next. They're running in the background. You don't normally see them in the sense of the end user. But that code is shared among lots of different pieces of software, word processing, spreadsheets, you name it. They'll have lots of shared coding elements, which are shared because they're functionally useful. And as Winston puts it, guys who write code are lazy. Why reinvent the wheel? I'll just reuse this module, this coding module. Depending on the context, I can layer specific functional elements on top of that. But the respective fraction of my total program, maybe a lot of it, maybe 75, 80% of it between a word processor and a spreadsheet will be shared. So he says, I see this pattern in software of dependency where some functional module will depend functionally on another module. That deeper module is shared among a number of programs. So what he did then was to say, well, what would happen if we compared universal common descent as a prediction to a dependency graph relationship? You would get very different patterns. Universal common descent predicting one pattern of distribution, dependency graph predicting a different pattern, where a dependency graph is much more congenial or fits better with an intelligent design framework. And then he comes up with a mathematical metric for assessing the relative strengths of these, where every time you have to add an assumption to common descent, it pays a penalty. Where you have to add an assumption to dependency graph, it pays a penalty. And then he assesses these in relative mathematical terms. It's a very technical paper. It takes a lot of thought to understand, but I think he's on to something. So I hope that made sense.
Mm-hmm.
He comes at the biology from the perspective of someone who writes software, who understands how it works. So he's applying functional insights from complex systems that we ourselves have developed and saying, can these possibly illuminate how genes and proteins are shared among the whole tree of life?
Mm-hmm.
I think the paper is going to go through a series of evaluations. Right now, it's being critically evaluated by a larger community. And I'm persuaded that Winston's core insight will survive the critique and is bound to be very fruitful.
Winston's actually in the audience with us today. He's a regular. Let me ask you a question.
We should have let him explain his own idea. If he was there, why did I do it?
Well, maybe we'll have him in the future, the webinar on this topic. But I just want, I know you've only got a few minutes left, but just to pick your philosophical mind, the question relating to common ancestry and the cosmological argument, actually. This is actually an argument that a friend of mine came up with recently, and it seems like a really interesting argument. It's to do with the B theory of time, which is one of the main one of the main rebuttals to cosmological arguments, both the Kalam version and the Leibnizian version of the cosmological argument based on contingency, which basically says that it's kind of meaningless to talk about the beginning of the universe in the same way that you wouldn't talk about the beginning of a ruler being the first centimeter, right? And so you could try to hold that the universe is just a brute fact; it just exists necessarily, or that you don't need to talk about the explanation for the origins of the universe's beginning. Now, my friend Vladimir came up with this really interesting argument. The argument runs as follows: 1, if evolutionary biology is true, then change must have occurred insofar as there has been a genuine development of species. 2, if B theory is true, then all species are equally old or ageless as a static block itself. 3, therefore, if B theory is true, then no development of species has genuinely occurred. 4, therefore, if B theory is true, then evolutionary biology cannot explain the complexity of developed species. As such, if the atheist is to accept the only plausible argument against Kalam, that is the B theory of time is true, apart from defending it against seriously destructive objections to endurantism and perdurantism, he also must abandon his only defense against the apparent design of species. Pick your poison.
Wow, that's wild. I do think it's interesting if you look at the lineage, if I can use that term, the lineage of evolutionary theory. It comes out of a theistic or even a Christian cradle where you have a genuine arrow of time, right? Mm-hmm. A finite arrow. A happens, B happens, C happens, and so forth. You get this unfolding. And actually, the original Latin meaning of evolution is not our current understanding, but rather a directed unfolding, which was one of the reasons that Darwin was hesitant to use the word in the origin. His own term, as you know, was descent with modification. The word evolution in the first edition only occurs once, the very last page of the book. So I think a genuine asymmetric linear progression of time where complexity accretes by natural processes lies right at the core of the modern understanding of evolution. And timelessness sits very uncomfortably with really any post-Darwinian conception of evolution. So I actually have run out of time.
Yep.
I'd like to do another one of these in a couple of months if you'll have me back and reprise the second part of my ontogenetic depth presentation. But thanks to the audience for tuning in. And next time, if Winston's in the audience, he should summarize his own idea.
And actually, he said in the chat, I always like to see how other people explain my idea to see how the telephone game has applied to it.
Well, I hope I did a reasonable job, Winston, if you're still there.
All right.
Take care, everybody.
Well, have a great night. See you later.
Thanks. Bye now.